Yeah, thank you very much for the invitation. And I really would have hoped to come to the meeting and to the speakers' dinner yesterday night, but that's the virus. Yeah, so I'm very happy to tell you about Biobank from our point of view, and that point of view is like we absolutely need it for our research. I hopefully can show you some very cool ways that we can use both body fluids and also tissue. So this is actually cutting edge research that I'm showing you today, and the one story is just on bioRxiv, and the other story is not published yet. But I hope it can actually change the way we can use tissues from the biobanks, or how we can use them to greater advantage. So this work is both from my group in Munich, where I'm now, and also from the group in Copenhagen at the NNF Center for Protein Research. So here are the two places where our groups are. And yeah, here in Munich, we are at the Max Planck Institute, and in Munich, we are at SUN faculty. So let's see if I can get a pointer here. Yeah. What we do is proteomics. So we're looking at the proteins in these samples. And just to refresh you, we would start with some sample of anything basically that contains proteins, such as here cancer tissue. And then we need to extract the proteins from that, and that we can do in a streamlined fashion in robots these days. So that's symbolized by this pipette tip here, where we would actually do all the sample preparation. And then we have here two platforms that we're using. So either this more well-known thermo Orbitrap platform for doing the proteomics, and then this here one time of flight, I'll mention it, and that gives us a very, very high sensitivity that we need for single cell work that I'm going to talk to you about. And then once we have that data, then it's the same as in all other areas of omics, that you need a lot of bioinformatics to actually make sense of this data. So, and then over the years, we've used this a lot in cell biology to ask basic questions in cell biology, cell signaling, and so on. But the last 5 years, we're really switching a lot to clinical applications, and we hope the next phase will indeed be to roll it out on a population scale if we get a chance. So, so lots of technology development, which I will not talk so much about. And here it's more conceptually. So once we have this technology, what can we actually do with this proteomics technology? And if we look at the cross-section of a cell, we can look not only at the expression levels of the proteins, but in principle, we can look at where are the proteins, like where in the cell are they, or in the extracellular matrix that's indicated here. And we can also study dynamic processes as we do something to the cells, or as we get tissue from, let's say, patients over different times of their disease course. And then here we can also look at post-translation modifications that's indicated here. So whether the proteins are in an active state or not in an active state, for instance. So just to show you, illustrate how far this technology has come. So this has nothing to do with biobanks, but this is a large-scale study we published half a year ago in Nature. And what we did here is that we took the proteomes of 100 different species, and we measured them. And then, so these were all species where the genome was already sequenced. But what we could then add was what does the genome actually get translated to? So we could say what are the most abundant proteins for all these 100 different species, from archaea, bacteria, to mammals. So and that was done with a fancy workflow that's illustrated here, and it actually also doubled the number of experimentally verified proteins in one go. So that's just by illustrating how far the technology has come. And Now I start with the first story, and that's body fluids, specifically the plasma. So it's been clear for a long time that it would be a good area for proteomics to get into to study the proteins in the plasma. So there was already a false dawn, I would say, 20 years ago, when people said they could diagnose ovarian cancer, for instance, from blood. But then more recently, you may have heard this story of a drop of blood with um, a company in San Francisco, but that turned out to be fraudulent. So the idea that we will use, that we want to use proteomics or some sort of measuring of the proteins in the plasma, that's already been apparent for decades, but that we can actually do it with mass spec-based proteomics, that's very recent. And why is that? As you can see this here on the upper left, that's because of this so-called dynamic range problem. So we need to measure, so we have proteins like albumin in our blood that transports things in the blood and other transport proteins here. And then on the other extreme, we have signaling proteins like cytokines, and between them there's over 10 orders of magnitude in concentration. So that's analytically very difficult. So I can liken it to measuring the weight of a jumbo jet by itself, or jumbo jet with an apple put on the wing, right? So it's really, you need very advanced technology for that, and that's why it's taking so long. So then here's a bioinformatic analysis here. So what are these proteins actually doing? So for instance, we have acute phase proteins in the top range of the abundance, and then we have the cytokines that are already mentioned here. And in the middle, we have a lot of tissue leakage proteins. So they have, many of them have no business to be in the plasma, but they're just being spilled into the plasma. And if an organ like the prostate is somehow damaged, or the heart, then it will spill the specific proteins of that organ into the plasma, and that's then elevated, and we can say maybe this person has a risk for heart disease, or they've had a heart attack, or they may be on the way to prostate cancer. So so in these days of omics, and specifically genomics, you may think it's all done by genomics nowadays, but that's not the case. So this is from the biggest hospital in Munich here. We also work together with, and we've tallied that for this review that you can see in molecular systems biology. So of the people that come into the system at all, 77% of them get a lab test, and then out of those millions in this one hospital here per year, and the same as at Ries Hospital also, then a plurality of them get tested for proteins. So that's the blue wedge here. And another big wedge is small molecules. So that's metabolomics, and that's also done by mass spectrometry. So if everything, if we had developed our technology, then I would argue that all these lab tests should actually be done by mass spectrometry instead of by ELISA as they're done now, because one of the advantages would be that it's much more specific, and the other advantage would be that you could measure everything at once. So you would get not only the level of one reagent, but of hundreds all at once. So that's our goal. So this is this review where we also go into why this hasn't happened before, and you can get this poster. So you can go to this molecular systems biology website. And we have developed, with Philip spearheading that on the Munich side, we have developed a workflow where we just take a finger prick of blood here, then use robots to work this up, and then have the mass spectrometer, and then come to this protein pattern in your blood, and that's so that we had here. And then we have done a number of studies. We've looked at how does the plasma proteome change on weight loss, and bariatric surgery, and we also done this for newborns, of 2 early born children, and a wide range. But the story that I'm going to talk to you about today is liver disease, and this is done by Lili Neo in my lab in Copenhagen. and also done with a network headed by the next speaker, actually, Tom Hansen, where we look at the liver. And so a large proportion of the population has fatty liver. So this here is alcoholic fatty liver, but it can also be a fatty liver just because of obesity. And this can then go on to steatohepatitis and then to fibrosis and cirrhosis. And then this is irreversible here. And people actually, and there's very high mortality. So you would like to catch this at earlier stages. And right now, there are decades-old enzyme tests for the plasma, or there are ultrasound-based imaging tests where you look at how stiff is the liver. But this tends to be a little bit late, and so this is a large unmet need. So what we did is we looked first in NAFLD, so that's Lily here and Philip. So Lily is soon going to defend her PhD, actually. So here we looked at Danish cohorts, like you can see here, and these were relatively small, but we nevertheless found, using this technology here, we found potential biomarkers, and then we actually also verified them in mouse models. So so that's what we already did, and now it's a large study that's comes from Odense, and it's more than 600 people now, and then we have a fancy workflow here with a lot of quality control, robots, and then we do this by a particular mass spec method that's called data independent acquisition. So we get not so many missing values. And then in this case, we used, or Lily needed 26 days of measuring time. And then we do machine learning and a lot of data analysis here. So, and the good thing was here that we had actually, for a large number of people, we had actually also the tissue and so paired samples from tissue and the blood. And then that could be the first question. So when, so what is the abundance of potential biomarkers in the liver itself and in the plasma? So you can see there are two populations. So some are more abundant in the liver itself, and some are abundant in both. And what we're interested in is then if we have potential biomarkers, then would they could be going up just in the tissue, or they could be going up both in the tissue and also in the circulation. So again, they would be spilled into the circulation. And what we see here, we have actually both. And the C-reactive protein, that's sort of the most famous marker for cardiovascular health, that actually is a very, very good marker of liver disease also. We actually argue a better marker of liver disease, perhaps. So some of these are used in the clinic now, and now we can say, are they coming more from the liver, potentially also which cell type in the liver, and we also have many examples of things that build up in the liver, but they're not shed in the circulation. And what's more is that you can see here in the heat map, so we have patients, so this cross-sectional, not the same patients, but we have patients in each of 4 categories of fibrosis. And then we have their tissue proteomes here, and then we have the same 4 stages in the plasma. And we can see examples of proteins that go down in the liver and go down in the plasma across the fibrosis stages. But then we also have ones that go in parallel, or even we have some that go up first in the liver and then they, with a delayed, some delay in the fibrotic stage, they come into the plasma. This, of course, is very exciting because you can be a little bit more mechanistic about what do these biomarkers actually mean. And this is the thing I'm most excited about, because working with a group in Odense with Alexander Krau and his team, we then took the best test that they have, which they also partly developed, and then compared it to the mass spectrometry. So this is a bit complicated, but this is this usual receiver operator curve for how well can we actually predict the disease cause, or how much does it correlate in the first instance with the fibrotic stage. And it turns out that we actually right now on par or even better than the best tests that are out there for the early stages of fibrosis. So we can tell from the proteome profile what are they, do they have this fibrotic state or on the road to fibrosis or not, because you can do this just from a finger prick, and the technology is getting rapidly better. I think this is very exciting, and we are actually now looking into that. We can do this. We need to replicate this in larger cohorts, but we are thinking about, can we actually roll this out for a Danish population at risk, so the cost will not be prohibitive. But of course, it's going to take a number of years, because this all needs to be verified, and there need to be people that want to do this and to pay for it also. So this is very exciting, and the good thing is this is generic. So once it's for liver disease, it will be the same test almost, because we're just looking for the proteins in the plasma for metabolic risk, or more generally, or maybe heart disease, and maybe also late presenting cancers as we increase the depth of the technology. So this is then our vision. So we would do a lot of these studies, which we are doing already, and then we would get a big matrix. So what do these patterns mean? And that's illustrated here. So liver disease or heart disease, or what is your reaction to a drug? Will it even work? And this will be lifestyle, for instance, vegetarian versus some Western diet lifestyle, and these are quality controls. So that's one use of these studies and biobanks. And then the other one is very new and very hot, I would say, and that's can we actually use tissues and go down to the level of individual cells or individual cell types. So it's illustrated here. You may have seen this before for deep single cell sequencing, which there's a lot of now, but we want to do it with proteomics. So this is the argument here that when you do this in bulk and you mix all this, like like shown here in a blender, then you would get an average of everything. But if you could pick out the individual cell types, you would get, this looks more like a banana, and this looks more like a pineapple, and so on. So we would be better able to assign changes in the proteome, in this case, to what cells they are coming from. So this project here is headed by Andreas and Marvin in Munich. So that's the technology part, that we need to increase the sensitivity of our mass specs. What we are doing is either laser microdissection, or what I'm showing you now is FACS sorting into 384 wells, and then this symbolizes the HPLC part, and this symbolizes the mass spectrometer, or is a cartoon of the mass spectrometer. So we work with these both companies. So this HPLC is actually a company that's in Odense, so it's spearheading this. So this is the Evosep, and I also have to say that I'm an indirect investor in this company in Odense. So that's disclosure of possible conflict of interest. But basically, this is a very robust system. And what's new here is that we push out the liquid at a very, very low rate, and that dramatically increases our sensitivity, and is still a very, very robust system as shown here. So then the other part is the mass spectrometer itself, and there we're working very close together with this Bruker company, which is a German and American company, and we need to increase the signal of this instrument, and it's shown here. And this is shown here that we indeed can do this, so it's 5 times higher signal. And then this is the result already. So when we then FACS sort either 0 cells or 3 cells into one well, or two cells into one well, and so on, then we indeed can see, even from one cell, we can see 1,000 proteins. So we're quite proud of that, and that gives us now a handle on looking at specific cell types. So this is a quantification. So you see a plot of bulk proteome, 200 nanogram against bulk proteome. So that has a good correlation. But that's even the case still when we go to 8 cells only. And that's even the case, very interestingly, if we go to 1 cell. And that, as you may know, if you're working with transcriptomics, that's not the case, not for technology reason, but because the cell doesn't need every message for every gene to be present there all the time. So it's inherently noisy from a shotgun noise point of view. So what we've seen here is that single cells actually have a stable proteome. So we've done a lot of experiments now, whereas they don't have a stable transcriptome, and that's in the nature of the biology. So now what can we do with this? So we have a history here of looking at tissues, and in the context of cancer or metabolic disease or neurodegenerative disease. And there we look at archived patient samples, and you can actually use, by proteomics, you can use FFPE material because proteins are quite tough in contrast to RNA. So in this paper here, for instance, in working together with a group in Chicago and Slengi, here we've cut out the tumor area here, microdissection, or we've cut out the stroma surrounding the tumor, and it turned out that the marker that one could do something with was actually in the stroma, which was good news because the stroma is genetically stable, whereas the the tumor, of course, evolves when you do something to it. So, so that was before, but now the new ideas, and that's spearheaded by Andreas Mund and Fabian Koscher at the CPR in Copenhagen. Now we want to use the tissue, and then we want to use deep learning for both segmenting the cells and then also classifying them. So that's our idea. And then we also have something that I don't have time to talk about, but that's a bioinformatic tool called the clinical knowledge graph, where we put all this in and put it together with all the knowledge that's out there in terms of publications and databases. So this is a large team effort that also involves the group of Peter Horvath here for artificial intelligence, and also a group from Leica, because we had to, or they had to adopt the microscopes that we use. So this is a typical slice from a tissue. This is salivary gland, so it looks quite beautiful. And now the first task is to use deep learning, so artificial intelligence, to segment the cells to just that the computer can see where they are. So that's here from the Horvath group. And now it's segmented the cells based on markers for the plasma membrane and so on the morphology. And this is an example for fallopian tube from ovarian cancer. So it's drawing now the outlines of the cells, and then it's also going to use machine learning to actually cluster the cells. Either we teach it, what are the different cell types here, or we ask it, please give us n number, for example, 8 different classes of cells on the basis of morphology or antibody staining or whatever you want. So this has happened here in the example of the salivary gland. So this is morphological classifiers, and then it gives them pseudo colors, or it gives them colors based on, we asked it to find 5 different classes here. So we don't actually know what these classes are. And then this is the the thing that happens next. So first, yeah, we also need to find landmarks in the actual slides so that we can transfer it from one imaging to another imaging, and also to the microscope that's actually going to cut them out with a laser. So that's illustrated here. And this illustrates here how how the software then finds different classes of cells and groups them together. So it's actually very smart about it, and it uses the latest tricks in deep learning, so you don't need like millions of examples. So this is an example here how that would actually look like. So laser microdissection. So it's first drawn the outlines that you can see in yellow here, and that would then would actually with the laser cut it out, and it can be quite fast, so a few seconds per cell. So it could actually do 30,000 cells per day. And then, and this is something that's called biological fractionation. So if we now not analyze the whole tissue, but each class of cells separately, then we will get the most abundant proteins or the characteristic proteins of that cell type. We get them to show up in high abundance, right? So here we have the protein abundance rank here shown. So this will be most abundant proteins and least abundant protein, and then a characteristic protein for this type of cell would be the most abundant protein once if we cut out only that cell type, and also these are other characteristic proteins for that cell type. And this is another example of exactly the same. So here we're looking at melanoma. So again, the AI has classified cells here and marked them in color. Yeah, and then we get the specific proteins that have been annotated in the literature to be specific to melanoma, and they are then cut out. And we can go one step further. Nothing tells us to stop at just the cells. We can also cut out the nuclei, and then we will get processes, biological processes, when we then analyze it, that are characteristic for the nuclei or the cell or the cytosol. So that's illustrated again here, and classifies them into 6 classes in this case. And then the proteome will also cluster into the same 6 classes, and we can see what proteins are characteristic of that class. So it turns out this is in cell culture, this is actually the proliferative status of the cell. And we can now, so in closing here, that we can put this together with all kinds of imaging developments now. So for instance, this is showing that we can extract these features in 3 dimensions, and then we need to cut them out. So classify them in 3 dimensions, and even if we see any kind of interesting thing from your biobank samples here, for instance, what is this here? What are the cells trying to do here? When they have this contact, this can also be cut out and analyzed by proteomics in the future. So, and then we hope that this will be a decision support. Yeah, that's the last slide. Decision support for clinicians. Okay, so thank you for your attention, and I hope you found this interesting. And yeah, so and we hope to be in contact with biobanks and measure them through eventually. So thanks for your attention.